Hello everyone, welcome to the fourth session of Physics Talk 2021. I will start by introducing myself. I'm Zubaida Karim Juthi, junior student in physics discipline, Khulna University. It is a great pleasure to introduce our speaker, Marzuk Ahmed. He is currently working as a research assistant in Biophysics Research Laboratory. Without any further ado, I would request our speaker to start his talk. Thank you.
Thank you everyone. Assalamu alaikum. I have completed my BSc degree from Khulna University and have completed my MSc degree from Bangladesh University of Engineering and Technology, that's BUET. And now I am working as a research assistant at Biophysics Research Laboratory. Actually, my research, today I will present my research experience in my MSc research and as a research assistant as well. Actually, the topic of today's presentation is theoretical and experimental aspects of human cell-like giant vesicles. I would like to share some theoretical aspects, not everything, but I have noted earlier. The outline of the presentation is was the introduction part. At the introduction part, I would like to present some basic things related to my research. That is biomembrane. That is actually the membrane of the cell, human cell. And after that, I will present the estimation of bending modulus of these vesicles. That is the bending modulus of the membranes of the vesicles. And after that, I will talk something about electroporation technique. That is a non-thermal minimal invasive technique to destroy the cancer cells. Actually, by this technique, actually the membrane of the cell is destroyed, which is a cancer cell. And after that, I will talk about nanoparticle induced rupture of vesicles. We know that there are some interactions of nanoparticles with the membranes of the giant vesicles. Vesicles means the artificial cell. It is an artificial compartment that is synthesized in our lab, biophysics research laboratory. So at first, it is very important to know that what is biomembrane. Biomembrane is a fluid structure that there are many constituents here, as like protein, the oligosaccharide, then cholesterol, phospholipid. But the main constituent here, we can see in this figure, in this illustration, that the main constituent here is the lipid, and it is a very complex structure. So to avoid the simplicity, we only consider the lipid structure here. So the protein or cholesterol that's are eliminated from here. So here is the vesicle. Vesicle is an artificially prepared spherical compartment. That is artificial. We can say it as like artificial cell. So actually, this a cell or a human cell or animal cell is actually the compartment that is inside the compartment. There are some fluids, and outside the compartments there are also some fluids. And here is the bilayer. We can see a lipid bilayer here, that these are the lipid molecules, and when these lipid molecules forms a spherical shape like this, and this is called the vesicle. This is artificial cell. And here this is a at the right side of the top, this is the confocal microscopic image of cells. We can see the ring here. This ring is actually the membrane of the cell. Yes, and this is the lipid bilayer. So at the bottom here, this is also, there is also a phase contrast microscopic image of a cell. This bright ring here is actually the lipid bilayer, and the interaction of the nanoparticles or other things with this cell is related to this lipid bilayer. So we consider here only the lipid molecules here. There are various types of vesicles. Mainly, there are two types of vesicles. One is unilamellar vesicles, that is composed of one lipid bilayer, and another one is multilamellar vesicles. That is, there are two, three, or four or more lipid bilayers here. And the unilamellar vesicles are actually three types. Number one is small unilamellar vesicles. The size of the small unilamellar vesicle is 25 to 50 nanometer, and they are called the SUVs. And number two is the large unilamellar vesicles. The size of the large unilamellar vesicle is 50 nanometer to 1 micrometer. And this is our point of interest, which is giant unilamellar vesicles. The size of the giant unilamellar vesicle is greater than 10 micrometer. So this is our actual interest, because the size of the real cell is ranges from 10 to 50 micrometer, and the giant unilamellar vesicle is greater than 1 micrometer. So we can consider the giant unilamellar vesicle as the artificial cell here. So the GUVs, these are called GUVs, that is giant unilamellar vesicles. This is called a mimic of the real cell. Now, it is important to know the impact of salt concentration on the membrane and on the membrane of the real cells. As for example, the salt concentration can affect various things in the cell, that is transport of various materials through the membrane of the cells, and the nutrient level of the cells would not be appropriated if the salt concentration changes from the equilibrium value. And this can also affect the ionic imbalance of the cells with the change of the salt concentrations. That is, the salt concentration means the internal fluid, or the salt, that is sodium chloride salt. Sodium chloride, there is sodium chloride salt solution inside the membrane and also the outside of the membrane fluid. So if the salt concentration changes, that is, for example, in the inside or outside of the membranes, these things can be occurred. If the salt concentration changes from a specific level, the cells can be ruptured easily, so the cells are break down. Then there are various impacts on the cell if the salt concentration changes. So therefore, it is important to know how the ions of the salt in solution are distributed near a charged membrane. Here is the ionic distribution of the membrane. At that upper figure, we can see this is a negatively charged lipid bilayer, that symbolizes by here is the negatively charged lipid bilayer. Actually, this bilayer, this lipid bilayer, composed of two types of lipid molecules. One lipid molecules are neutral, and actually it is 60% of the lipid molecules are neutral, and 40% of the lipid molecules are negatively charged here. So here is a structure of the lipid bilayer. This is negatively charged lipid. If the ionic concentration changes inside or outside of the membrane of a cell, then some things can be occurred. That is, if the salt concentration increases, then there are more sodium ions that are inclined here, that are attracted to the negatively charged lipid bilayer. So if the concentration of the sodium ions are increases, then the negatively charged lipids are more attracted to them. Then the electrostatic interaction between these negatively charged lipid molecules decreases. And if the electrostatic interaction decreases, then the bending modulus of the membrane decreases. That is, the cell can change its size. We will see later. That is, we will see later in detail. And if there are electrostatic interaction decreases, then the size of the cell also decreases. And if the electrostatic interaction increases, then the size of the cells increases. So when the sodium ions are increased, increased here, so the electrostatic interaction decreases between the negatively charged lipid molecules. And if there is an increment of the negatively charged lipid molecules here, so then there are more negatively charged lipid molecules in this region, then there are more electrostatic repulsion between them. So the electrostatic interaction increases here, then the size of the cells increased. Here is a graph here. So the if at here is the in x-axis, this is the distance, and the right y-axis is for surface potential, and left y-axis is for ionic concentration of the membrane. So very close to the membrane, the ionic concentration of sodium is very high, and when the distance from this lipid bilayer increases, then the ionic concentration of the ionic concentration of the sodium ions are decreases. We can see from this figure. And the ionic concentration near a charged membrane can be expressed as Boltzmann equation. The C plus is for positive charge, and C minus is for the negative charge, and psi is the electrostatic potential, and rho is the density of the ion in the solution. And this is some basic things, that is Debye length. Debye length is the measure how long the electrostatic interaction can persist. And this is the simplified form of Poisson-Boltzmann equation here. Now I would like to share my MSc thesis. My MSc thesis was entitled "The Effects of Salt Concentrations on the Size Distribution of Giant Unilamellar Vesicles." I have performed this task by two manner. That is, one is changing the salt concentration in buffer from 12 to 412 millimolar for a fixed surface charge density of the membrane. Here I have fixed the surface charge density of the membrane, but changed the salt concentration in the solution. And the solution is called the buffer. The buffer solution I have used is PPS buffer, that is composed of 10 millimolar PPS solution. This PPS is can be found in the powder form, and the pH of the total buffer is 7, and EGTA is also can be found in powder form, and here is the sodium chloride salt. After mixing these components with these concentrations, that is PPS is 10 millimolar, EGTA concentration is 1 millimolar. EGTA stands for ethylene glycol tetracetic acid, and here is the sodium chloride solution. And this buffer is similar to the fluid inside the cell or outside the cell. So we have prepared this buffer here. And to understand the effects of the salt in the membrane extensively, I have fixed the salt concentration in the membrane, and then changes the surface charge density. That is, I have changed the negatively charged lipid molecules here. Actually, the equilibrium percentage that I have said earlier is 40% negatively charged lipid molecules are in the membrane. But I have changed the charge of the membrane, that is here is for 10% to 90%. That is 10% to 90% negatively charged lipid molecules in the composition of the membrane. Um, I have prepared the artificial cell that is called vesicles, and I have noted, I have said that the giant unilamellar vesicles are the mimic of the cells. I have prepared the giant unilamellar vesicles in our laboratory by natural swelling method. It is a well-known method to prepare the artificial cell. The lipids are used here. Two types of lipid. One is negatively charged lipid, that is DOPG, and another one is electrically neutral lipid, that is DOPC lipid. This is a schematic diagram of preparation of the vesicles. At first, the mixture of these two lipids are taken in a glass vial. That is the total volume is 200 microliter lipid solution, and I have dried the lipid by nitrogen gas, and there a thin film of the lipid. This is actually these lipids are dissolved in the chloroform, and when we dry it by nitrogen gas, the chloroform is eliminated and dry lipid film can be stayed in this glass vial. Then for fine dry, we have kept it in a vacuum desiccator for 12 hours. Then the second stage is the prehydration stage. These dry lipid molecules are prehydrated by 20 microliter milliQ. This milliQ is ultra pure water. Sorry, it is the nano pure water. MilliQ is the nano pure water. That means there is no ions, no particles, nothing in this. Only here it is composed of hydrogen and oxygen. This in this prehydration stage, this dry lipid film is prehydrated by 20 microliter milliQ water, and the temperature here is 45 degrees Celsius, and we have prehydrated it by for 8 minutes. And number 3, this stage is called incubation stage. In this incubation stage, the buffer solution that is called the Pipes buffer. The buffer solution is added to the glass vial, and also 0.1 molar sucrose is also added to this glass vial, and this is the solution. Then I have kept it at 37 degrees Celsius for 3.5 hours in an incubator. The incubator temperature is 37 degrees Celsius. Here I have fixed the temperature at 37 degrees Celsius because the natural temperature of our body or of our cell is 37 degrees Celsius. Then after 3.5 hours, these artificial cells are created. That is this circular shape we have seen here. We can see here in this inside this solution, these circular shapes. These are the artificial cells, and here is a phase contrast microscopic image. I have used a phase contrast in an inverted phase contrast microscope that was imported from Japan, and the model of the microscope is IX73. And from this microscope, we can see this type of images, and these circular shapes here are the artificial cell, and we can see the bright we can see many bright walls compartments, and these walls are actually the membrane. Then in the left side below, we can see many small lipid aggregates here, and there are also some larger lipid vesicles. To eliminate these smaller vesicles, actually these smaller vesicles, the size of these smaller vesicles are less than 10 micrometers. So we have to avoid them because our point of interest is 10 micrometer is greater than 10 micrometer. So to purify these lipid vesicles, I have used 10 micrometer pore sized filter. This is an filter paper, and it is an schematic diagram of the purification process. So there is the flow is controlled by a peristatic pump, and the flow rate is 1 ml per minute. When these lipid molecules from this is a syringe, actually these are the syringe. When from this lipid solution, this lipid solution is from the right syringe to left syringe, and here is the filter paper. Then the vesicles, those are less than 10 micrometer. They are uh, uh, they uh, they can pass through this filter paper, but which vesicles are sizes greater than 10 micrometer, they cannot pass through this filter. So now we can collect these vesicles from under this filter paper. We can see here in the schematic diagram. After the filtration technique, we have found this type of vesicles here. This is also a phase contrast microscopic image. Here there are very small amount of smaller vesicles, and here is the most of the vesicles here are greater than 10 micrometer. Now from various phase contrast microscopic images like that, here I have shown the in A. This is a phase contrast microscopic image of the giant vesicles. From this type of images, I have counted the size of the vesicles manually, and the size of the vesicles can be counted by a software that is provided that was provided with the microscope. The software is cell sense entry software. This software can with the help of this software we can measure the size of these vesicles manually. So we have calculated about 300 to 400 vesicles and their sizes, and after gathering the information about their size distribution, I have prepared a histogram that is here in B. This histogram is for 330 number of vesicles. So here and from these histograms, we can find the average size of the vesicles for a fixed salt concentration or for a fixed surface charge density. Here, this is for here the salt concentration is 12 millimolar, and the surface charge of the membrane is x equal 0.4. This means there is 40% negatively charged lipid here. So from this condition, this is I have shown here a specific condition, and that is the C is stands for the salt concentration, that is 12 millimolar. And from this size distribution with the log normal distribution, this is a log normal distribution, and from this these lines are for this dotted line is for the log normal distribution, and from this distribution curve we can find the average size of these vesicles for a fixed condition. or for a specific condition. And this is the log normal distribution, and this solid line here is for the bending modulus of the membrane. And we have found this bending modulus. We have developed a theory. We have a Russian collaborator. His name is Victor Levadny. He is from Russian Academy of Science. And he actually help us to develop the theories about these things, or in our other research. So this solid line is for measuring the bending modulus of the membrane. And we are the first research team. We have those who have found the bending modulus of the membrane theoretically from the size distribution of the vesicles. And this is the equation for this solid line. Here we can see that K bend, and this K bend is actually the bending modulus of the membrane. And from this distribution we have found the bending modulus of the membrane. And the unit of the bending modulus here is in energy unit, that is kBT units. And the electrostatic term here is K electrostatic. Here the electrostatic equal gamma x square divided by cube square root of the c cube. Here x is stands for the stands for the surface charge density of the membrane, and c stands for the salt concentration of the membrane. And we have changed both the salt concentrations and surface charge density of the membranes here. And then we have found the d average. D average is the average size of these cells, the average size of these cells. And here is a relation between the average size of the cells and the bending modulus of the membranes. This is the theory of the size distribution. Here the total system can be determined by the Helmholtz free energy. The equation 1 is stands for the Helmholtz free energy. And here are some mathematical calculations here. Then finally, after simplification and other calculation, we have found equation number 7. This equation, that is Nm, this is a function of the dm. Dm is the diameter of the vesicles, that is the diameter of the cells. Then from this equation, now we can found the bending modulus of the membrane. Here L is L is a constant, and K bend is the bending modulus of the membrane that we have found from this equation. And D frequency and DM, these are the fitting parameters. And the L here L equal N5 by 4 pi. Here N is the number of the vesicles, or the number of the cells, which is our point of interest. And phi is a volume fraction of the vesicles. And we have found these things, that is bending modulus of the membrane, that can be defined as the energy required to deform a membrane from its natural curvature to some other curvature. When the bending modulus is larger, there are large diameters vesicles can be formed, and when the bending modulus is smaller, then the size of the cells can be decreased. This phenomena can be clear from these two graphs. At the left side, there is the change in the average size of the vesicles, and in the right side graph we can see that when the surface charge density of the membrane increases, that is the percentage of negatively charged molecules, that is negatively charged lipid molecules in the membrane increases, the size of the cells increases. And also the bending modulus of the membrane increases. And that is there is also a proportional relationship exists between the average size and bending modulus of the vesicles. And we have published these works in Physical Review E, at 2020, and this is my MSc research finding. And after that, I would like to share my experience as research assistant. And at first, I would like to share my experience on electroporation. That is irreversible electroporation. Irreversible electroporation is actually a non-thermal technique that can destroy a cancer cell by this is the electric field can be applied in a localized cancer cell. And actually the electroporation technique is they have two types. That number one is reversible electroporation and another one is irreversible electroporation. In the reversible electroporation process, when an electric field is applied to the membrane of the vesicles, the membranes can reseal after sometimes. But in the case of irreversible electroporation, the membranes can be permeabilized. That means the membrane of the cells can be destroyed. So the cancer cell can be destroyed by irreversible electroporation. Here are some applications that is for cancer treatment, tissue ablation, and drug delivery as well. And this is a schematic diagram here where we applied the electric field. This is a microscopic slide where we apply the electric field to the vesicles. And this whole thing is an experimental design of the irreversible electroporation technique, that is IRE technique, for the pore formation of the vesicles. And in this image, before application of the irreversible electroporation, the membrane we can see the cell here is completely spherical. But after the application of the electric field, the membrane is destroyed and the whole the cell is destroyed totally. That can be seen is the image below. So by the application of irreversible electroporation, we can destroy a cancer cell like this, and this is totally localized. So the other neighboring cells cannot be affected with this type of application of the electric field. Here are some theoretical aspects of the irreversible electroporation process. And the irreversible electroporation, here actually the electric tension is applied to the vesicles, and with the increment of this tension in the vesicles, the vesicles can be ruptured totally. This is the equation of Maxwell stress tension of the vesicles, and here Vm is the transmembrane voltage, that is 1.5 Re. R here is the radius of the vesicles, and E is the applied electric field. So the total tension of the membrane, that is when the electric field is applied, the total tension in the membrane can be calculated by this equation. Epsilon m is the permittivity of the membrane. And in the left side figure, this is the probability of pore formation after the application of the irreversible electroporation. That means after the application of the electric field on a cell. Here are these are our experimental data. These circles, the circular the shape, these are for one type of lipid composition, and the squares are for another type of lipid compositions. There are many types of data here, and the solid lines here are for theoretical fitting for this experimental data. So we can see that here sigma E is the tension that is applied on the membrane by application of the electric field, and in the y-axis this is the probability of pore formation. That means pore formation in the membrane or the destruction of the membrane. So when the tension is increased to some extent, that is then the probability of pore formation in the membrane is also increased. And in the right side graph, we can see the rate constant of pore formation. The rate constant of pore formation means the in which rate the pore formation occurred in the membrane. So the rate constant of here is denoted by the Kp. When the when the tension on the membrane increased, the rate constant of the pore formation also increased. And this is a simple overview of this thing. We have changed many things. That is, we have concluded cholesterol in the membrane. Then we have changed many things in the solution, and we have changed the various composition of the lipid molecules in the membrane, and we have found very interesting results. And we have published these works that are related to irreversible electroporation in European Biophysics Journal, and Chemistry and Physics, Chemistry and Physics of Lipids Journal, and various journals. So there are some publications that I have denoted here. Now, here is the nanoparticle induced deformation and pore formation in the membrane. Nanoparticle can bind with the membrane of the vesicles, and they interact with the lipid molecules. Then they also can form pore in the vesicles. That means the nanoparticles can also be used for treatment of the cancer cell, or the drug delivery to a localized cells. The drug delivery to a localized cell, actually this phenomena is when and at first and a nanoparticle is inclined to the membrane of the vesicles, then that form a pore in the membrane. Then a targeted drug that is inside the, that is inside of the inside of the nanoparticle can be delivered through the pore of the membrane. Then the drug delivery can occur inside a cell. Here is a schematic diagram. That is how the nanoparticle can bind with the membranes. And the right here is the right side graph. In the right side graph, we can see that in the y-axis, this is the compactness of a cell. The compactness of a cell, that means when how the cells can be transformed to a spherical shape, to other shape, or the deformation can be occurred. In A, we can see that this is the time 2, 14, 26, 31, 46. These are the change of change of the vesicles with time. At 2 seconds, the cell is completely spherical, but with the change of the time, the shape of the cell has been changed, and at the 56 minute, the cell is completely deformed, and we can measure the how how the cell can be deformed is by measuring the compactness of the cell. The compactness can be measured by calculating its area, its diameter. These two things are related by calculating the compactness of a cell. So we can see that at B, uh, when the with the increment of the time, the compactness of the vesicle is increased. So is increased. So this means that the nanoparticle bind with the membranes of the cell, and at the figure C, we can see the average compactness. Average compactness means the compactness of various types of cells. Various types of cells means the composition of the cells. That here we have also changed the composition of the lipid molecules, that are neutral and negatively charged lipid molecules. Uh, and in the below, now we can see that with the time, the fraction of deformation increased in the vesicles. That means when a nanoparticle is binded to a membrane, then the fraction of deformation increased with time. Uh, and also with the increase of nanoparticle concentration in a solution, the deformation is also increased. That can be seen from this graph in the below, the second graph in the below, and we have published this work in Chemistry and Physics of Lipid journals in 2020. So this is an simplified overview of my total research experience, and thank you everyone. So if you have any questions, you can ask me. Thank you.
I had a question actually. In
Yes,
that
sure.
ionic distribution membrane slide, maybe it was your second slide.
Yes, yes.
Uh, okay.
This slide.
Yes, this one. Okay, so you have used a Boltzmann equation to derive this formula, actually, right? So here you have written down the formula for the linearized case. I wanted to ask that if what was the actually assumption for a linearized case?
The assumption for the linearized case here is that this is some mathematical calculation. We can calculate the ionic distribution is C plus is for the positively charged ions, that is sodium ions, and the C minus is for the negatively charged ion, that for the chloride ions. And we can calculate the ionic charge distribution by this equation here. This is denoted by the rho, and here E is the electrostatic charge, and by calculating this we can, here the linearized case is for the sine high, this is the sine hyperbolic function, and this assumption actually I would like to share some calculation here that can be expressed in the calculation with these equations. This is some fundamental equations, and when I have not shown here it in detail here.
Okay, that's okay.
So sorry, I cannot help you to understand this in detail. So it is it will be good if I can share this by some mathematical calculations that can be derived in detail.
No, it's not necessary actually, but it's okay. I would like to know what would be the condition of the temperature for the linearist case actually. And
temperature here all the temperatures are 37 degrees Celsius.
so for the linearist case, we do not have to change the temperature to a certain amount or something, right?
No, we don't have to do that. This here is K is the Boltzmann constant, and T here is in every case the value of T is 37 degrees Celsius, because in the case of a real cell, our general body temperature is 37 degrees Celsius.
Thank you very much, Marzuk, for your excellent actually presentation and your very significant research result. Actually, these are findings are very already you prove that these works already published in a very good journal. These works are actually experimental. You did, right?
Yes, sir. This is totally experimental. Jyoti, you have asked here. I can understand this slightly your question, because for in the linearized case, we have used here the this charge distribution. We have calculated actually the electrostatic potential, that is denoted by the psi. We have calculated the electrostatic potential here, and for we have calculated the electrostatic potential for various conditions. that is related to the membranes. And for some values of this electrostatic potential, now we can find that this sine hyperbolic function that is related, that is beta psi, is similar to only the beta psi. So so this is the linearized case. This actually this is only the we have taken into account the electrostatic potential. Then we have calculated this in the case of the linearized case actually.
Actually, that was my question. What should be the condition of the electrostatic potential?
Um, the condition for electrostatic potential is here are some negatively charged molecules, that is lipid molecules in the region of the membrane, and we have changed the this proportion of the negatively charged lipid, and I have told that I have changed the negatively charged lipid from 10% to 90%, and there are various values. For example, 10% negatively charged lipid molecules, then 40% negatively charged lipid molecules, then 55%, 70%, and 90% negatively charged lipid molecules. And here is the function is the negatively charged lipid molecules. So when the negatively charged lipid molecules increased to some value, the electrostatic potential can be changed. And also now with the change of the salt concentration, that the salt concentration in the buffer here, and that is the change of the concentration of sodium and chloride ion in the buffer, that can also affect the electrostatic potential. These are the two parameters to calculating the electrostatic potential.
Okay, thank you.
So you said that you're using your findings to destroy cancer cells. So did you actually simulate or artificially created a cell with cancer?
Hmm. Actually, this is not a cancer cell, but this is related to the cancer cell, that we can consider this as cancer cell. Mainly, the target is to destroy the wall of the cell, and this wall is composed by the lipid molecules, or this is called the membrane, or this is called the lipid membrane of the cell. And if we try to destroy the lipid membrane of the cells, then the cell can be destroyed easily. And the cancer cell is also composed of these lipid molecules, and also the wall of the cell is composed of these types of lipid molecules here. So if we can destroy this artificial cell with these conditions, here for example, this is a very high electric field. We have changed the electric field from 300 to 840 volt. And here the frequency is 1.1 kilohertz here. So with these conditions, and this is applied to a cell for a millisecond duration. So there is no hamper can be occurs to the cell with this little amount of time. So if you we have applied the electric field to the membrane of the cells, and we have found that this can be destroyed with some conditions. And we have checked this for various types of cells. Various types of cells means various types of lipid compositions. We have used various types of lipids here. We have changed the amount of negatively charged lipid, and we have used here cholesterol. That is cholesterol. We all know that cholesterol is also is a part of the membrane of the cell. And we have also included here some cholesterol. Then we have applied the electric fields, and then we have observed the phenomena. And with various conditions, we have observed the phenomena to destroy the structure, or to ablate the total cell. And this is this is a very hot topic for cancer research now, but clinically it is not applied yet, but the research is ongoing in this field.
Yeah, I have a question. So artificial cell means it's like a nanorobot, and it can be applied on the cancer cell. And my question is this: how do you control this artificial cell?
Control means in which case?
To destroy the cancer cell. So if there are a few number of the cancer cells, so
yes, actually the total process is here we can see the microscope, and we have prepared a glass slide. Then we have prepared a small compartment. The size of the small compartment is 300 about 300 microliter. So now we have pushed the solution with the artificial cells to that compartment. Then we have inserted some electrodes. The electrode is actually gold coated electrode, and we have bought this from China. This gold coated electrode, it is not available in our country, that we need in this research. So we have inserted the electrodes. The distance between the two electrodes is about 25 millimeter. Then we have inserted it to the compartment that we have prepared in the slide. Then we kept it at under the microscope, and then we apply the electric field to the cells with these electrodes. And we have developed a low cost power supply device that can be controlled in some specific case for our experiments. Then we have applied the electric field to the cells, and in real time we can see what happens to the cells with the application of the electric field by the microscope. And I have said that here the microscope, here also a software is provided with the microscope. With this software we can see the real-time phenomena which can occur to a cell when we apply the electric field to the cell. This is the total experimental setup actually. Is it your question, or
yeah, thank you for the answer.
I have another question actually. How about if I apply the external magnetic field rather than electric field, and what is the physical actually process here? Interaction with the electric field and that cell actually.
Sir, actually here the interaction is related totally to the solution that is called the buffer solution. And here in the buffer solution there are some positive and negatively charged ions, that is sodium ions and chloride ions, and it is also dependent on the surface charge of the membrane, that is the charge of the lipid molecules. When the charge of the lipid molecules in the membrane increased, the rate of the ablation of these cells also increased. And it is also related. It is the electric field is here related with these two things. That is the ionic distribution of the buffer and the surface charge density of the membrane.
Yeah, I understand. So actually here the magnetic field will not work.
No sir, we haven't applied this, but I think the magnetic field cannot affect this.
Oh, I see. Although that is actually the charged particle, right?
Yes sir.
So magnetic field there should have some effect.
Some effect. I think here the effect.
People are using such kind of thing.
I think, sir, we haven't find any report to apply the magnetic field. But I think if we applied the magnetic field, there are some the change in the compactness of this artificial cells. But the rupture of the cells is may
I
be
mean,
difficult with the magnetic field.
I mean, whether they have any research actually,
No,
using
sir, we don't have find any things related to the magnetic field application.
microwave field. How about if you give the time dependent electric field?
Mm, um, Sir, here the time dependent electric field means we here apply the
Alternating current. You produce here the electric field by using a just anode and cathode, right?
yes, sir, just a anode and
The
cathode.
alternating current. So what happened here?
Alternating current that there may be if we use the alternating current, we cannot apply it locally. So the neighborhood cells that are good cells that can be affected with the alternating current.